This meeting is being recorded.
Acha, so good afternoon, everyone, and welcome to this presentation on current and future advances in genetic testing and systemic autoinflammatory diseases. My name is Md Nahian Anwar. I'm from Dhaka University, and today we will be discussing the latest developments in genetic testing and their potential implications for clinical practice. Systematic autoinflammatory diseases are a group of rare genetic disorders characterized by uncontrollable inflammation throughout the body. According to the article author, genetic testing is essential for diagnosing and guiding treatment for these diseases. Currently, genetic testing for systemic inflammatory diseases typically involve analyzing a specific set of genes known to be associated with the conditions. However, this approach has limitations, as not all cases of systemic inflammatory disease are caused by mutations in these genes. In recent years, there have been significant advances in genetic testing and technology that offer new possibilities for understanding the underlying causes of systemic inflammatory diseases. For example, article author describes how RNA sequencing can be used to identify changes in gene expression that may be involved in disease involvement. Additionally, epigenetic profiling can reveal changes in DNA methylation patterns and may contribute to disease progression. Finally, whole genome sequencing offers a comprehensive view of a patient's genetic profile and potentially identifying new disease-associated genes or variants. Article author also noted that functional genetic testing, such as assessing immune cells function or cytokine production, may provide a more direct understanding of the molecular mechanisms underlying systemic autoinflammatory diseases. This type of testing can identify immune system defects that may not be apparent through genetic sequencing alone and may lead to more targeted treatment options. Now, before we start reviewing the whole article, we have to know a few words. The first word is inflammation. Inflammation is a very recognizable word. Inflammation usually means a localized physical condition in which part of the body becomes reddened, swollen, hot, and often painful, especially as a reaction to injury or infection. Inflammation is very important for us in order to fight against foreign pathogens. However, when inflammation happens against our own body particles, it is called autoinflammation in such autoinflammatory diseases. So here is the inflammatory spectrum where you can see what is autoimmunity and what is autoinflammation. Today we will be basically talking about the left wing of this figure because this article consists of only autoinflammation. So autoinflammation is the activation of innate immune response without an apparent cause, which ultimately is bad for our health. Now there are a few more words, which is mosaicism, digenic inheritance. We will go there, and autosomal dominant and autosomal recessive. These words are new, and we will talk about it furthermore in the journal when we talk about it. Another word is de novo. De novo is specifically more responsible with mosaicism. It is a Latin term that means anew, from the beginning, or afresh. And in silico means a study which is conducted by computer modeling. Now let us start. The methodology. The methodology of this article is a review article. It is a literature review article which analyzes and synthesizes existing research and clinical applications. The sources of existing researches are various databases such as PubMed, Medline, and Embase. So this is the methodology of this article. Now let's jump into introduction and abstract. So as we all know that autoinflammatory diseases are mostly monogenic, means the shift in a singular gene or in an allele of a same gene.
Hello. Can you interrupt you?
Yes, of course.
You are not changing your slide. You just show your first slide, but you are not changing it.
I am changing in. Maybe it is not showing. Is it not showing?
Uh,
Okay.
Yeah, right.
Okay, let me start again. I didn't know it was not showing. Select Windows screen, current future and testing, about. Okay, so is it showing now?
yes, right now it is changing.
Okay. Thank you, sir. Okay, So de novo in Latin term, it means anew or beginning or fresh, or in silico is a study conducted by a computer model. So we will talk about pure autoinflammation, not autoimmunity. Autoimmunity is more based on adaptive immunity, which is T cells and B cells, but we'll talk about only innate immunity, which is responsible with only cytokines, various cytokines, and natural killer cells. So now let's go to methodology. The methodology is this is a review article, a literature review, which analyzes and synthesizes existing research and clinical applications. There is not much statistics provided in this article. Sources of the author, which authors used, are various databases such as PubMed, Medline, and Embase. Now let's jump into introduction and abstract. So mostly autoinflammatory diseases are monogenic. Monogenic means a shift or change or mutation in a single gene, which is non-allelic or biallelic, or change in biallelic genes in both of the genes of the same allele. So they are called monogenic. Digenic is our next slide. We'll talk about it. So about 60% of patients with suspected monogenic systemic inflammatory disease are still molecularly undiagnosed. So 60% of patients are having no treatment because they're undiagnosed. Diagnosis of systemic autoinflammatory diseases is hampered by paucity, which is the scarcity of clinical criteria and functional diagnostic tests. Until today, to this date, over 30 genes responsible for systematic autoinflammatory diseases are found. There are a variety of modes of inheritance. Now, inheritance, as it is a genetic disease, inheritance is a big part here. We will talk about Mendelian law of segregation and other things. So let's go to the next slide, which is the next most amazing topic of this journal, which is monogenic autoinflammatory diseases and mode of inheritance. So first we'll talk about the mode of inheritance. The most common and classical mode of inheritance is autosomal dominant and autosomal recessive inheritance. We all know that we have sex chromosomes, and most of our body or our somatic cell chromosomes are called autosomes. So when autosomal dominant or autosomal recessive genes are passed down from parents to their children, they often get the auto-inflammatory diseases. An individual with an autosomal recessive genetic disorder must carry biallelic pathogenic variants in the same gene. It is transmitted mostly from parents. For example, it is a mevalonate kinase deficiency, which is known as MKD, which is caused by biallelic mutations in the MVK gene. Now, the second one is mosaicism. Mosaicism is a very amazing and new thing. It is usually regards with atypical and unexpected mode of inheritance. It is a de novo mutation. De novo, we have learned in the first slide that de novo is a new. So in this case, new conformations of chromosomes, new conformations of genes can be seen. It is mainly responsible for somatic mutations, means changes in the body, in the outer body parts. It happens post-zygotically, means after the formation of zygote. Mosaicism is caused by de novo mutations that occur post-zygotically, so-called somatic mutations, which result in two genetically distinct cell populations within a single individual. For example, pathogenic somatic mutations in NLRP3 were shown to cause neonatal onset multisystem inflammatory disease. So this is not a single system, multi-system are hampered through this inflammatory disease. It is also known as CINCA, chronic infantile neurological cutaneous and articular syndrome. If the somatic mutation is also found in other types of cells, including gonadal tissue, it is called germline or gonadal mosaicism, and the mutation has potential to be passed on to the subsequent generation. Germline mosaicism has been reported in patients with Blount syndrome and with tumor necrosis factor receptor 1, respectively, and in a patient with Eneral family card domain containing protein 4 associated hemocytosis lymphocyte disease. So there are lots of disease in mosaicism. However, here there is both of these instance has the same common thing, which is both of them are monogenic, a single change. So we will talk about another thing, which is digenic inheritance. In here, it is a non-Mendelian inheritance. As we know, Gregory Johann Mendel had three laws of inheritance: law of segregation, law of independent assortment, and law of dominance. So in digenic inheritance, it is not usually shown that exactly how that person has inherited a such gene which is responsible for autoinflammatory disease. But it must be demonstrated by a family-based segregation analysis. Without that, we cannot usually detect a digenic inheritance, because sometimes, most of the times, people are asymptomatic in digenic inheritance. Digenic inheritance manifests if mutations occur simultaneously in two distinct genes, in two distinct cells or two distinct tissues. The digenic pathogenic variants are usually identified in proteins that are part of the same multiprotein complex. A classic example of digenic systematic autoimmune diseases in the group of proteasome-associated autoinflammatory syndromes, also known as chronic atypical neutropenic, which is in short is called Kendall. A Kendall syndrome it's another disease. So digenic and oleogenic inheritance has also been reported in other types of initially characterized systematically autoinflammatory diseases. The oleogenic type of inheritance is in the continuum between monogenic and polygenic inheritance, whereby a phenotype results from coherence of several low-frequency variants. Allele frequency in this case is 1 to 5%. The allele frequency is not low enough to explain association with rare disease, and these variants are often identified in unaffected parents. So the parents mostly in digenic inheritance are unaffected, and they show no symptoms at all. They're asymptomatic, but the children, they get digenic inheritance, which does not follow the Mendelian law of inheritance. It is a non-Mendelian inheritance. However, the law of segregation can be amplified here to demonstrate the certain pathway of digenic inheritance. Now, there, besides these three, there is another thing which is multifactorial inheritance and the contribution of environmental and epigenetic factors. Most known systematic autoinflammatory diseases are monogenic diseases, as we discussed before, but there are also a number of multifactorial and complex systematic autoinflammatory diseases such as Beckett's disease, systemic juvenile idiopathic arthritis, and Crohn's disease. Epigenetic factors are defined as heritable changes in gene expression that do not affect the genomic DNA sequence. The DNA sequence remains unchanged and do not affect, but they synthesize different kind of proteins, which are epigenetic factors. These complex genetic diseases are not suitable for genetic diagnosis and genetic counseling, as these risk variants or haplotypes are also found in asymptomatic persons, which means such conformation of DNA sequence can be found in a fully normal person as well, where they are not causing a disease. However, they can cause disease in some people, as it is a multifactorial inheritance. There is no singular factor responsible for such kind of inheritance. Because epigenetic changes are less stable and easier to modulate than DNA mutations, they serve as integrate environmental signals and to mediate interactions between environment and the resulting genomic output. The best understood epigenetic mechanisms comprise DNA methylation, histone modification. Histone is a pro-inflammatory cytokine. Histone modifications, remodeling of chromatin, and non-coding RNAs, and some of them have been associated with pathogenesis of systematic autoinflammatory diseases. Genetic testing methods for autoinflammatory diseases. The most used genetic testing method is the Sanger sequencing. Sanger sequencing still constitutes the mainstay of molecular testing of SIDs. This method is well suited for low throughput laboratories for which NGS means next generation sequencing approach would not be profitable. So small labs or small laboratories with not much of access to modern machineries, Sanger sequencing is a very good option, and most of the people use Sanger sequencing to test the genetic testings of autoinflammatory diseases. This method is it is recommended for patients with a clear clinical diagnosis. So a patient with clear clinical diagnosis that, for example, a patient is diagnosed with Synca or any other autoinflammatory diseases. So a certain clinical diagnosis is needed to do Sanger sequencing. We cannot do uncategorized autoinflammatory disease genes in Sanger sequencing. Sanger sequencing can also assist in the confirmation of a biochemical diagnosis in patients with NKD or DADA2. Both of them are autoinflammatory diseases, and thereby contribute important information to genetic counseling. Furthermore, Sanger sequencing is also indispensable in confirming variants that were identified via NGS method, which means Sanger sequencing is also indispensable in confirming variants. We have to identify through next generation sequencing and then confirm it is a variation of a previous gene. We have to use Sanger sequencing. Nevertheless, performance and efficiency of Sanger sequencing in SADs is low, except for patients from founder populations with FMF and NKD. Both are autoinflammatory diseases. Now, next is NGS approaches, which is next-gen approach, next-gen sequence approach. So next-gen sequencing panels are current method of choice for patients who fulfill criteria for several possible diseases or with ambiguous phenotype. So the difference between Sanger sequencing and NGS approaches is Sanger sequencing, you need a confirmed diagnosis, but for NGS approaches, it can be a randomized disease or a person can show multiple symptoms of multiple autoinflammatory diseases. So NGS approaches usually confirm the which autoinflammatory disease is that certain person or particular person is diagnosed with. It can be also complemented by bridge PCR and droplet PCR. And next comes the whole exome sequencing. Exome, it's mainly mRNA sequencing, the coding regions of our DNA. And the mRNA is sequenced here. And there are many mRNAs which are responsible for mainly exons. Then we all know there are two parts of mRNA, exon and intron. Introns are non-coding regions of a mRNA, and exons are coding regions of a mRNA. We only code the exon here. Now the next thing is whole genome sequencing. Whole genome sequencing is a very good option compared to the whole exome sequencing. It is to capture the human genome in an unbiased manner. It can detect disease-causing variants found by conventional approaches. Whole genome sequencing also has the potential to identify deep intronic variants. Intronic variants means which are non-coding yet can be or can play a role inside an autoinflammatory disease. And other cryptic mutations. Cryptic mutations means hidden mutations which are not yet transcribed or synthesized but will be non-coding regulatory regions and therefore increases the diagnostic yield. Moreover, the genome-wide uniformly distributed coverages allows a much more reliable identification of CNVs as compared with whole genome sequencing. An additional important advantage of whole genome sequencing is the opportunity to periodically reanalyze the genome-wide data for recently discovered and reclassified variants. Despite the emerging evidence on effectiveness and benefit of whole genome sequencing, it is still mainly used for discovery of novel disease genes in research setting. It has no medical setting yet because it is too costly and there is not enough professional manpower to handle such a genome sequencing. So it is mainly used for research purpose, though it can be a very good option in the medical purpose. Now, we'll talk about classification criteria for genetic variation.
Md Nahian Anwar, আমি একটু ইন্টারাপ্ট করি, আপনার স্লাইড মুভ করছে না।
স্লাইড আবারও মুভ করছে না?
হ্যাঁ, এখন ঠিক, হ্যাঁ ঠিক আছে, এখন ঠিক আছে।
আচ্ছা আচ্ছা, thank you. Classification on criteria for genetic variation. There are quite a lot classifications. It is based on actually by which evidence we prove that the genetic variation is happening. The first is evidence based on computational predictions, which we talked about in the first slide, is in silico. Every movement or every translation of gene, we model it inside a computer and we see the results and predict a result. And the second is evidence based on population data. Population data are an important line of evidence to support the pathogenicity of identified variants. The allele frequency of severe disease-causing alleles is expected to be rare in the general population, and the frequency of sequence of greater than 5% in any population is considered standalone Binning classification, which means if a population has greater than 5% frequency of an allele which can cause autoinflammatory diseases, it is known as a very, very dangerous thing. And that population can soon be growing and be fully of autoinflammatory diseases. Evidence based on functional studies. Functional study means mainly laboratory experiments. First one is the computer predictions. Second one is the population data, which is kind of a survey. And the third one is laboratory experiments. So in laboratory experiments, for example, although many autoinflammatory diseases are mediated by cytokine interleukin 1 beta, interleukin 1 beta is a pro-inflammatory cytokine. Pathogenicity of novel matched LPR and PYD domains, these three are autoinflammatory diseases. Domains containing protein 3 variants should be assessed by measuring cytokine production in patients' serum samples, as the serum levels of interleukin 1 beta are functionally irrelevant. A more reliable functional assay is to measure the interleukin 1 beta production in supernatants of stimulated peripheral blood cells. So in the periphery, the blood cells, if they contain more interleukin 1 beta, that means that person is most likely to be in an autoinflammatory disease. Evidence based on mode of inheritance and segregation. Because monogenic disorders are expected to be inherited in a Mendelian fashion, as we talked about, the evidence of segregation of a specific variant in a family can also be useful for interpretation of its clinical significance. For instance, the occurrence of a variant de novo, which means a new, is considered strong evidence of pathogenicity if the several criteria are met. Confirmation of maternity and paternity is very important. We have to look towards the mother and father genes of a certain child to understand exactly what kind of inflammation is going on. Presence in a gene associated to a disease that is consistent with phenotype, parents must definitely be unaffected. So if a parent is affected, they are not advised to get child, but if the parents are not affected, then a child has more chances of being affected. Mostly autoinflammatory diseases, if the parents are affected, childs are not affected. In contrast, inheritance supporting the evidence from unaffected parenting considered strong evidence that the variant is benign. The application of the described criteria relies heavily on accurate and correct phenotypic evaluation of all included family members and can therefore be influenced by factors such as penetrance, expressivity, and age of disease onset. So the disease reflection in which age is very important here. Now we will talk about what slide or something. Now we will talk about limitations of this particular article. So there are mainly two limitations. Might miss the deep intronic variants. Intronic variants are the variants which are not directly related to the coding, but it's a non-coding region of mRNA, but can influence the coding. So might miss deep intronic variants that affect protein translation and expression. However, to date, there are no deep intronic variants or variants in regulatory elements described for systematic autoimmune diseases. And most of the times, one particular test, we have talked about many tests here. So one particular test may not be enough to confirm a certain gene. For DADA2, several pathogenic deletions and duplications comprising one or several exons of ADA2 gene. DADA2 is caused because of ADA2 gene. So have been described. Depending on size of the CNVs, Sanger sequencing alone would not be sufficient for their detection, and more elaborate laboratory techniques such as multiplex ligation dependent probe amplification, digital droplet PCR, ddPCR, or next generation sequencing approaches are needed. Now, for conclusion, the number of molecularly characterized systematic autoinflammatory diseases has grown rapidly over the past 20 years and has been fueled by development of new sequencing technologies. Many more genes are still unknown, and over 50%, that is almost the half of patients, are mutation negative. They don't show any mutation, but they are diagnosed with autoimmune diseases. So autoinflammatory diseases. So these genetic factors are very exciting to study. In conclusion, genetic testing plays a crucial role in diagnosing and guiding treatment for systematic autoinflammatory diseases. The latest advances in genetic testing technology offer new possibilities for understanding these complex diseases and may ultimately improve patient outcomes. However, as with any medical technologies, we must carefully consider the ethical implications of these new approaches. Now, autoinflammation and autoimmunity is the second largest source of worry for microbiologists and biotechnologists of the future. The first is, of course, antibiotic resistance, but the autoimmune thing is growing rapidly throughout the last 20 years, and it is a relatively very new science and a new branch of science we have to study. So, as we all know, the type 1 diabetes, which is diagnosed very early in a child, type 1 diabetes is also an example of an autoimmune disease in that our T cells, our body's cytotoxic T cells, attack the cells of islets of Langerhans in our pancreas, and we don't produce insulin. Our insulin producing cells are binded by T cells. So that is just a singular example. There are inflammations in the brain, there are inflammations in the heart. So inflammation, autoinflammation is going to be the next big thing after antibiotic resistance if we survive that. So my remarks about this article is it is a pioneering research, of course. It is one of its kind and it's a start. And a newly branch of science needs more research in it, more researchers in it. But the scarcity of numerical values in this article is very much noticeable. There should be more statistics provided, statistical values, and it requires extensive study. Further researches should be made. Now, thank you for your time. The one last thing that is the food for thought. The father of modern art, Francis de Goya, he get an autoimmune disease before his art went scary. In 1972, he was diagnosed with a very mysterious disease at that time. After that, he went deaf, he could not hear, and his arts were turned into very scary things. Maybe his mind turned into scary things. So until today, there is an American Association of Autoimmune Diseases. They are predicting that Francis de Goya was a victim of autoimmune disease. Now, thank you for attending this presentation on current and future advances in genetic testing in systematic autoinflammatory diseases. We hope you found this information informative and thought-provoking. We look forward to continued progress in this exciting field. Finally, we'll have a question and answer session where you can ask any questions you may have and share your thoughts on the topic. Thank you, everyone.
Thank you. Thank you so much, Nahian Anwar, for your nice presentation.